The fluid circulating through lymphatic vessels and lymph nodes is known as lymph. And the lymph mainly has plasma proteins, and the cells predominantly found in the lymph are lymphocytes. And as the lymph absorbs the digested fat, we can see a milky white fluid which is known as chylus. And the lymph also acts as a channel for distant metastasis or for the spread of infections. uh, it can spread the cancerous cells, then microorganisms and foreign bodies. Lymphatic system starts with the formation of lymph capillaries. The lymph capillaries will unite to form lymph vessels, and these lymphatic vessels are the tributaries of a large terminal lymph duct. So we have lymph vessels and lymphoid tissue as the component. Lymph vessel includes lymph capillaries, lymph vessels, and lymphatic ducts. Then the lymphoid tissue. We know there is diffuse lymphoid tissue and dense lymphoid tissue. In the diffuse variety, we have malt mucosa associated lymphatic tissues like Peyer's patches. In appendix also, we have the lymphatic tissue, and the secondary lymphatic organs like lymph nodes, spleen, tonsil are there. The primary lymphatic organs are thymus and bone marrow. So these are all included in the lymphoid tissue. Lymph capillaries begin as blind-ended sac. So this is the lymph capillary. This entire is lymph capillary, and these are the cells of the tissue in which the lymph capillary begins as blind-ended sac, and these are the endothelial cells of the lymph capillaries. So we can see that the endothelial cells of lymph capillaries are more permeable than the blood capillaries. They have larger space, so they will permit entry of larger lipid molecules from them. And the most of the lymph capillaries, they carry lymph towards the lymph node, and the lymph node receives this lymph through afferent lymph vessels, and through the hilum of lymph node there is emerging of efferent lymph vessel which will carry the lymph away from the lymph node. And most of the organs have lymph capillaries in them. Again, important for MCQ, you should remember the lymph capillaries are absent in epidermis, cornea, articular cartilage, hair, nail, splenic pulp, bone marrow, choroid, internal ear, meninges, brain, spinal cord, liver and lungs. So these are the organs where the lymph capillaries are absent. Small lymph capillaries will unite to form large lymph vessel, and these lymph vessels will unite and they will form the ducts, lymphatic ducts. On right side we have right lymphatic duct and left side we have the thoracic duct. These, if we see histologically, the large lymph vessels are similar to the veins. They also have the three coverings of tunica intima, media and adventitia. Only the difference between lymphatic vessels and vein is that the elastic fibers are present in all the layers of lymphatic vessel, and the smooth muscles are present in tunica media as well as tunica adventitia. And in the larger lymphatic duct like thoracic duct, the smooth muscles are arranged longitudinally in the tunica adventitia. When we will study the histology of lymphatic system, we will repeatedly come across the term lymphatic nodule or lymphatic follicle. So let us see what is this lymphatic follicle. Now you can see these two structures are lymphatic follicles. These are concentration of lymphatic cells. These are well circumscribed but they are not capsulated. These are either spherical or ovoid masses. And the lymphatic follicles are present in all the dense lymphoid tissues. So try to recollect the dense lymphoid tissue. These are spleen, lymph node, then tonsil. These all have lymphatic follicles in them except thymus. The thymus does not have lymphatic nodules or lymphatic follicles. And the lymphatic follicles are of two types. So these are primary lymphatic follicle and these are the secondary lymphatic follicles. The primary lymphatic follicles or nodules have mature lymphocytes in them and there is absence of germinal center. And here the cells have larger nucleus and there is very thin rim of cytoplasm. So nucleus stains blue. That's why it is densely stained with blue color because the nucleus is larger compared to cytoplasm, and they are present before birth, and then after getting exposed to the antigen, the primary lymph nodule is converted into secondary lymph nodule. So the primary lymphatic nodule is mainly formed by mature lymphocytes and they have larger nucleus and these are densely packed. Then this is the secondary lymphocyte. Now we will see the difference between primary and secondary. So in the secondary lymphocyte, the secondary nodules, they show germinal center. So if you compare it with the primary follicle, you can see this is the pale staining area. It is stained with pink color, eosinophilic. So this is the germinal center. Germinal center lies in the center of the lymphatic nodule and it contains large immature lymphocytes known as lymphoblast and plasmablast. So in the germinal center we have immature lymphoblast or plasmablast, and the cells of the germinal center stain lightly. Here the nucleus is smaller compared to the cytoplasm and so it stains pink. It is surrounded by small cells with dense nucleus. So these are the peripheral densely packed lymphocytes. So this zone is known as mature zone or corona, and the central part has follicular dendritic cells that help in proliferation of lymphocyte. So now we will again revise. The basic difference between primary and secondary follicle is that in the secondary follicle we have germinal center which is absent in the primary follicle. Primary follicle is made up of mature lymphocytes, whereas in the secondary follicle the peripheral zone has mature lymphocyte and in the central part we have the lymphoblast or the plasmablast. So initially when the organ is not exposed to any antigen, then primary follicles are present, and when the follicle is exposed to the antigen, then the lymphoblast and the plasmablast will appear. So there is appearance of germinal center. So is it clear? Primary follicle means the follicle is not yet exposed to antigen. That's why all mature lymphocytes are there, and once it is exposed to the antigen, then there is appearance of germinal center. So far we have studied the different cells of lymphatic system, and now we will study the dense lymphatic tissues one by one. So the dense lymphatic tissues are lymph nodes, spleen, tonsil and thymus. So first we will see histology of lymph node. So the lymph node is kidney shaped structure, and the main function of lymph node is to filter the lymph. So it has outer, so this is the convex surface of the lymph node, and there is concave surface. So the concave surface has the hilum part, and the lymph node receives lymph via afferent vessels. So these are all afferent vessels. They open in the convex surface of the lymph node. So these are all afferent lymphatic vessels which will carry the lymph into the lymph node via afferent lymphatic vessels. And after filtering the lymph node will drain the lymph into the efferent lymphatic vesicle, and these efferent lymphatic vessels are present in the hilum. They will leave the hilum. So it is kidney shaped. It has convex part and concave part. In the concave part there is hilum. Convex part provides opening for the afferent lymphatic vessels, and they will transport the lymph, and after filtering the lymph it is then drained out of the hilum through the efferent lymphatic vesicle present in the hilum part, and the lymph node receives blood vessels. They enter and leave through the hilum. So here the blood vessels, artery and vein are also present, and the efferent lymphatic vesicles are also there. Afferent and efferent lymphatic vessels have walls in them. In the section of lymph node, we can see histologically this is the capsule. So the lymph node is encapsulated. It is covered by capsule. Beneath the capsule, we have the subcapsular space. So this is the subcapsular space which is seen only in the lymph node. So presence of subcapsular space is the peculiarity or identification point of lymph node. After the subcapsular sinus, we have outer cortex. So this is all cortex. After that, we have the paracortical area, which is also known as thymus dependent area. The gland is separated from the capsule by the subcapsular sinus, and the afferent vessels open into the subcapsular sinus in which lymph is present. Now these are the trabecules. So the capsule sends septa within the substance or parenchyma of the organ, and these septa are known as trabecules. They extend from capsule in the substance of the lymph node, and they convey blood vessels. So we can see these are the blood vessels present in the trabecule. The paratrabecular space on either side of the trabecule, we can see this is the paratrabecular space, and it is occupied by reticular fiber, and there are interstices filled with the cells of primary lymph follicle. So again we will repeat, this is the histological slide of lymph node. So it is covered by capsule. Beneath it there is subcapsular sinus in which there opens the afferent vessel carrying lymph. Then we have the cortex in which lymphatic follicles are there. Then the paracortical area is known as thymus dependent area, which has more amount of T lymphocytes, and this part is the medulla. And the capsule sends trabeculae, and the trabeculae have blood vessels in them. They convey the blood vessel, and the paratrabecular space is occupied by reticular fibers. They form a supportive framework in which the lymphatic follicle is present. The supportive framework of the lymph node is formed by reticular meshwork, or uh, here the reticular cells and the reticular fibers form a supportive framework or supportive meshwork, and the spaces within this meshwork are occupied by small and large lymphocytes. The reticular tissue, now the same we can see in this diagram also. So these are all reticular cells, the reticular cells, and these are reticular fibers. So these are forming the supportive framework or the connective tissue that forms a framework for the lymph node, and within this framework there are spaces, interstices, which are filled with the lymphocytes. So these are the lymphocytes present in the interstices or the spaces between the reticular fibers or reticular cells. Up till now, we have seen the supportive framework of the lymph node. So it is mainly formed by the capsule, trabeculae and the reticular meshwork. Now we will study the parenchyma of the lymph node. So the outer darkly stained area of the lymph node, means from this point up to this, is the cortex. Outer darkly stained area is cortex, and the inner lightly stained area, this much, is the medulla. So this is lightly stained. Again, the cortex is divided into two areas. So the superficial nodular cortex shows presence of lymphatic follicles in it. So this is superficial nodular cortex which has lymphatic follicles. So in the lymphatic nodule we have central lightly stained germinal center which mainly has B lymphocytes which are in proliferatory stage. So there are B lymphoblast in the central part, and the peripheral part of this lymphatic follicle has densely packed cells which are lymphocytes. So this is outer or superficial nodular cortex. Then deep to this area is the paracortex area. It is known as deep cortex or paracortex. It lies between the nodular area of the cortex and the medulla. And in the paracortex, we mainly have T lymphocytes. T cells are present, and this area is known as thymus dependent cortex. Medulla is the innermost part of the lymph node. It is pale stained area. It is mainly formed by lymph cord and sinuses. So these are the medullary cords and these are the medullary sinuses. So it is formed by medullary cords, that they are the anastomosing cords mainly formed by B lymphocytes, macrophages and plasma cells, and the medullary sinuses are the spaces adjacent to medullary cords, and they contain lymph in them, and after filtration of the lymph it is drained into the efferent lymphatic vesicle through the hilum of the lymph node. So in the medulla part or the innermost pale staining part of the lymph node, we mainly have the lymphatic cords formed by lymphocytes and the lymphatic or the medullary sinuses, and these have the lymph which will drain into the efferent lymphatic vesicle. Now we will see how the lymph circulates through lymph node. So this is the lymph node. In the outer convex part of the lymph node, we have opening of afferent lymph vessels. So there are numerous afferent lymphatic vessels opening in the convex part of the lymph node. They will open into the subcapsular sinus. So this is the subcapsular space present between the capsule and parenchyma of the gland. The lymph enters into the subcapsular sinus. From the subcapsular sinus, it enters into the trabecular sinus. Trabecular sinus are present on at either side, or we can say they are adjacent to the trabeculae. These green colored structures are trabeculae. So adjacent to the trabeculae, we have trabecular sinus. So the lymph passes from subcapsular sinus into the trabecular sinus, and from the trabecular sinus it enters into the medullary cords. In the medulla we have the anastomotic cords. So the lymph enters into it and then it will leave the lymph node via efferent lymph vessel which is present in the hilum of the lymph node. Now there are three type of sinuses thus. First is the subcapsular sinus, then second is the trabecular sinus, and third is the medullary sinus. And two type of lymph vessel, afferent vessels which will carry the lymph to the lymph node, and efferent, it will drain or it will leave the lymph node. So there are afferent and efferent lymph vessel. The sinuses these three sinuses, these are filled with the reticular meshwork, and this reticular meshwork will filtrate the lymph, and after filtration the cellular debris and other antigens are removed, and the lymph gets filtered, and then it leaves the lymph node through efferent lymph vessel. Lymph entering into the lymph node gets filtered, so the cellular debris and other antigens are removed, and the other function of lymph node is production of lymphocyte. It maintains the lymphocyte count, and there is presentation of antigen to T cell, then activation and proliferation of B cell. The B cells are converted into plasma cells, and the plasma cells produce antibodies, and cell mediated immunity is maintained by T lymphocyte, and humoral immunity is provided by production of antibodies by B lymphocyte. The lymphocytes present in the lymph node enter in it via two sources, either by the afferent lymphatic vesicle or vessels, or they enter into the lymph node via high endothelial venules. So what is this high endothelial venule? It is the post capillary venule lined by cuboidal or columnar epithelium present in the paracortical region. It is the channel for the entry of T and B lymphocyte. Then these endothelial venules absorb water from the lymph, and the lymph nodes become palpable after they get enlarged in infection or a cancerous condition. Inflammation of lymph node is known as lymphadenitis. Here the lymph nodes are enlarged due to proliferation of lymphocyte, and the causes of lymphadenitis are either infection, most commonly tuberculosis, or spread of malignancies. Thymus is a primary lymphoid organ, and it is situated in the superior mediastinum. Thymus gland develops from third pharyngeal pouch, and the cells present in the gland, that is thymocytes, and the connective tissue network or framework is mesodermal in origin, means gland is endodermal in origin, whereas thymocytes and connective tissue in the gland are mesodermal in origin. The thymus gland grows till puberty. Thereafter, it will start regressing. It gets atrophied, and this process is known as thymic involution. Means till puberty, it will grow, but later on, its function is taken up by other organs. So, fibrofatty degenerative changes occur in the thymus, and it gets atrophied. And this process is known as thymic involution. Coming to the histology of thymus, so the thymus is covered by capsule. So the outer lining is the capsule. So from the capsule, septa or trabeculae will project into the parenchyma of the thymus, and it will divide the parenchyma of the thymus into lobules. So the septa is incomplete. It will divide the outer part of lobules, but it will not traverse completely through the parenchyma. So the medulla part, the inner medulla part is continuous with each other. So what we have seen, there is a capsule, there are septas. Septas will divide the parenchyma into lobule. Now the outer darker area of this lobule is cortex, and the inner lighter area is the medulla. The same you can see in this lobule also. So the outer darkly stained area is the cortex, and the inner lightly stained area is the medulla, and the medulla of different lobules continue with each other. And again, the point to remember is that thymus do not have lymphatic follicles or lymphatic nodules in them. They are made up of lobules which have cortex and medulla, and there are no lymphatic follicles or lymphatic nodules in them. Now we will see the cortex. So the outer or the peripheral darkly stained zone is cortex. In this cortex, we have the connective tissue framework formed by epithelio-reticular cells. So these are the epithelio-reticular cells which form a network or meshwork, and within these the spaces are occupied by the lymphocytes, and these epithelio-reticular cells are derived from third pharyngeal pouch. Also, the cortex has few macrophages which phagocytose the degenerated lymphocytes. The inner lighter zone of the thymus is known as medulla. It is lighter stained because it has less amount of thymocytes in it. And apart from thymocyte, the epithelio-reticulocytes are also there, and the peculiarity or the identification feature of thymus is presence of Hazel's corpuscles. So these are the Hazel's corpuscles present in the medulla of the thymus. Thymic corpuscles or Hazel's corpuscles are present in the medulla of the thymus. These are ovoid structures, and these are eosinophilic hyaline masses surrounded by type 6 epithelio-reticular cells. They secrete thymosin and thymopoietin, and they represent degenerated thymocytes, and their number increases with the age. Now we will see the cells present in the thymus. Epithelio-reticular cells are the supporting cells, or they are a form of connective tissue network, and they are derived from third pharyngeal pouch. These are known as nurse cells because they provide essential environment for differentiation and maturation of T cells, and they are of six types. First three types are present in thymic cortex, and last three types are present in thymic medulla. There are six types of epithelio-reticular cells. So these are the type 1 epithelio-reticular cells present beneath the capsule of the thymus. So they isolate the lymphocytes from connective tissue and from the capsule, trabecular blood vessels. And these cells are connected by tight junction or occluding junction. So these are the type 1 epithelio-reticular cells present just beneath the capsule. And what is their function? Their function is to separate these lymphocytes from the connective tissue framework which is formed by capsule trabeculum, and it also separates the lymphocytes from blood vessel. Now the type 2 epithelio-reticular cells, they form an isolated compartment for developing T cells and educate the T cells. We know that the immature T cells are not immunocompetent and they can destroy themselves. So an isolated compartment is provided to the T cells till they get educated or till they get immunocompetent. Then the type 3 epithelio-reticulocytes. These form functional barrier between the cortex and medulla. So this is type 3 epithelio-reticular cell. Just adjacent to the type 3 reticular cells, we have type 4 reticular cells which will form a junction between the cortex and medulla. So this is the corticomedullary junction. Then the type 5 fibers, they form the cellular framework in the medulla, and the most important are type 6 epithelio-reticular fibers which forms the Hazel's corpuscle, which is the identification feature of thymus. So in the central part we have eosinophilic hyaline degenerated mass of thymocyte, and it is surrounded by concentrically arranged type 6 epithelio-reticular cells which gives it onion peel like appearance. The cells present in thymus are mainly thymocytes, which are the T lymphocytes, and epithelio-reticular cells, which form supportive framework. Apart from these two cells, we also have few mononuclear phagocytic cells. We have fibroblast in the capsule, perivascular spaces, and medulla. and there are myoid cells. These cells will help to move thymocytes out of the gland. Functions of thymus are they secrete thymopoietin and thymosin. So the epithelio-reticular cells, they secrete thymosin and thymopoietin, and these two factors will help in maturing the uncommitted T cells so that they become immunocompetent. Then it provides an isolated environment for T cells until they get maturated. And thymus is essential for immunity in early life, means till puberty. After puberty, the function is taken by other organs, and thus the thymus starts regretting. A regression or atrophy of the thymus occurs after puberty. Coming to the blood thymic barrier. So this is the blood thymic barrier. We know that the immature T cells will mature and they will become immunocompetent in the thymus in an isolated environment. So these lymphocytes are present in an isolated compartment, and they are prevented from exposure to the antigens present in the blood vessel. So the components of blood thymic barrier, this is the blood thymic barrier, and its components are continuous capillary endothelium, then the thick basal lamella on which the endothelium is resting. It also has few pericytes. Then this is the perivascular space, and the tissue space contains tissue fluid. Then there is continuous layer of type 1 epithelio-reticular cells. So this is the blood thymic barrier. And why blood thymic barrier is present? Because the immature T lymphocyte till they become immunocompetent are present in an isolated compartment, and they are prevented from exposure to the antigens present in the blood vessel. Coming to clinical importance of thymus, so there are two clinical conditions, myasthenia gravis and DiGeorge syndrome. So myasthenia gravis is an autoimmune disease where autoantibodies are formed against the neurotransmitter acetylcholine, and there is weakening of skeletal muscle. And DiGeorge syndrome is a congenital syndrome, and it is due to deletion of segment of chromosome 22. And in DiGeorge syndrome, there is absence of thymus and parathyroid gland.